Humans are built around a biological paradox. We eat sporadically, often hours apart, yet our organs, especially the brain and heart, demand a massive, uninterrupted supply of energy every single second. To bridge that gap, our biology relies on two distinct fuel systems. The first uses fast-acting carbohydrates, stored as glycogen in the liver and muscles. But that supply is strictly limited. Without a fresh meal, these cellular glycogen reserves run dry in about 24 hours. This chart highlights the difference in scale. While glycogen provides a thin margin of safety, the vast column on the right represents our primary energy vault, adipose fat. This tissue holds tens of thousands of calories, enough to sustain a human for weeks. However, accessing that vault presents a chemical challenge. Lipids are hydrophobic. They physically repel water. Because human blood is a water-based medium, moving raw fat from storage to a working muscle is effectively impossible without a complex transport mechanism. Survival depends on the body's ability to transition its cellular machinery between burning these easy, fast carbohydrates and the specialized process of mobilizing those stubborn, dense fats. The direction of our metabolism is dictated by the most recent meal. Consuming carbohydrate-rich foods causes an immediate surge of glucose into the bloodstream. The pancreas responds to this spike instantly by flooding the blood with the hormone insulin. Insulin signals the tissues to prioritize glucose. As it binds to cell receptors, it coordinates a shift in cellular activity. It forces muscles to burn the incoming sugar for fuel, while simultaneously halting the breakdown of stored fat. Once the cells have sufficient glucose and the glycogen tanks reach their 5 to 6% limit in the liver, any additional carbohydrates are converted directly into fatty acids. To transport these water-repelling fats through the blood, the liver packages them into protein-coated spheres called very low-density lipoproteins, or VLDLs. This water-soluble shell allows the oily triglyceride cargo inside to move freely through the circulatory system. These particles travel until they reach adipose tissue. There, insulin activates enzymes in the capillary walls that split the triglycerides, allowing the fat cells to absorb and store the energy for future use. High levels of circulating insulin effectively prioritize storage, ensuring that excess dietary energy is converted and sequestered within the fat tissue. Hours after a meal, the process reverses. Blood sugar drifts downward, and the concentration of insulin in the blood vanishes. In this low insulin environment, especially during physical strain, the body releases counter hormones, glucagon, adrenaline, and cortisol. These hormones trigger a specific enzyme deep inside the fat cells, known as hormone-sensitive lipase. This enzyme breaks down stored triglycerides, releasing a flood of free fatty acids and glycerol directly into the circulating blood. These fatty acids bind to albumin proteins for transport, heading toward active tissues. Once inside the mitochondria, they are chopped into 2 carbon segments and oxidized to create ATP, the energy that powers muscle contraction. If carbohydrates are entirely absent, the liver initiates ketosis. It converts mobilized fats into water-soluble ketone bodies, which are small enough to fuel the brain's high energy demands when glucose isn't available. This transition represents an aggressive, hormonally driven excavation of stored energy, specifically designed to sustain the organism when food is unavailable. Fat functions as a communicative endocrine organ. As adipose cells increase in size, they produce more of the hormone leptin, which travels to the hypothalamus to signal that energy stores are sufficient and appetite should be suppressed. Evolutionarily, this system balanced energy through cycles of feast and famine. However, modern dietary patterns have effectively eliminated the fasting phase of the human metabolic cycle. Because of constant access to high glucose foods, the blood sugar spike rarely subsides. This keeps many people in a state of chronic, unending high insulin. Chronic insulin exposure maintains a persistent signal to store energy, making the mobilization of fat for fuel biochemically inaccessible, even when the body is in a state of relative energy deficit. This chart traces the resulting loop. Continuous carbohydrate intake drives insulin up, which traps energy in fat stores. As the rest of the body cannot access that fat for fuel, the cells signal for more energy, triggering hunger and leading right back to more carbohydrates. Over time, the body's tissues begin to ignore the insulin signal, a condition called insulin resistance. When cells no longer respond to the hormone that regulates their fuel, the resulting systemic breakdown, metabolic syndrome, leads to the global rise in obesity and cardiovascular disease. At this scale, the failure of internal regulation requires external intervention. Clinics and diagnostic centers have become a permanent fixture of the modern landscape to manage the fallout of this metabolic desensitization. Ultimately, once the body's autonomous fuel management system fails, we become reliant on external hormones. This vintage vial represents a century of medical effort to manually replace a biological transition that our modern environment has rendered nearly impossible to maintain. Body fat is a highly evolved survival engine. The difficulty is that its design, tuned for a world of scarcity, is struggling to function in an environment of constant abundance.